Simon has more than 25 years of experience developing and applying techniques to study local structure in materials using X-rays, neutron, or electron diffraction. He develops novel data analysis methods, including graph theory, artificial intelligence, or machine learning approaches. He earned his PhD in material science and engineering from University of Pennsylvania in 1992. After 13 years as a faculty member at Michigan State University, in 2008, he took up his current position as a professor of materials science and applied physics and applied mathematics at Columbia University, and physics at Brookhaven National Laboratory. Simon has published more than 300 papers in scholarly journals. He's a fellow of American Physical Society and Neutron Scattering Society of America, a former Fulbright and Sloan fellow, and has earned a number of awards, including the 2080 Warren Award of American Crystallographic Association, being honored in 2011 for contribution to the nation as an immigrant by Carnegie Corporation of New York, the 2010 Hannah Award of the International Center for Diffraction Data, University Distinguished Faculty Award at Michigan State, Thomas Osgood Undergraduate Teaching Award. He is also a section editor of Acta Crystallographica Section A, Advances and Foundations. He regularly chairs and participates in the reviews of major facilities and federal funded programs. Simon, the screen is yours.
Well, thanks so much, Paulina, and thanks so much for the invitation. It's really a pleasure to be here. This is actually a really exciting thing that you have going on quantum crystallography, and also I really like this idea that you have a non-quantum crystallography talk to balance it out. So it's really I'm very happy to be here. It's my privilege. I wanted to give, this is a talk that I actually gave the first time at ECM, and both saw it, and it's a little cheeky. It's kind of naughty, it's just an idea that's been floating around in my head, and then I have been slightly developing it, but I think it actually has legs and it's maybe interesting. So hopefully you'll enjoy it, even if you don't believe a word of it. So the title of the talk is "Do Materials Have a Genome? And if they do, what can we do with it?" So where does this come from? So I'm going to explain actually the origin of this, why we think about it, but it's really the origins go back to the Human Genome Project. So in the late 80s and leading up to 1990, the molecular biologists were actually having greater success in sequencing genes. They were starting to develop the kind of experimental technology to allow them to sequence genes. And they decided that this would be a really great moment in the history of mankind to have a kind of moonshot and say, okay, let's make a project that within the next 15 years will sequence the human genome. And when they set out on this journey, it seemed like they would never make it. And in the end, they succeeded in 2003, which was actually only 13 years after this was initiated. And there was a lot of funding thrown at it, but a lot of kind of creative work. It really motivated that community. And it was seen as a huge success. So that's the Human Genome Project. We can ask a question, what is a gene? Because we're crystallographers, maybe we don't know what a gene is. And I'm a crystallographer, I don't know what a gene is. But one thing a gene is, it's kind of a DNA molecule which contains these base pairs, and then there's a sequence of base pairs that form the gene, and the gene. So it's something to do with DNA, and it's something to do with the ability of organisms to reproduce from one generation to the next. It's like a memory of what came before. So that's one thing a gene is. It's something to do with molecular biology. Another thing a gene is it's basically a very large set of letters, basically. And these letters actually are the base pairs. So these letters indicate which base pair and what's useful here is actually the sequence of these letters. And a codon which codes for one protein is 3 letters. And this is actually very efficient coding, because if you had 4 amino acids, you could code for it with 1 letter, because there are 4 base pairs. And if you had 4 squared, which is 16, if you had 16 amino acids, you could code for it with 2 letters. But we have actually 26 or 27, I can't remember, of amino acids that are used in biology by organisms. And so you need 3 letters. In principle, that allows you to code for 64 amino acids, but we need to code for 26. So each set of 3 letters codes for 1 amino acid, and then the sequence of letters tells you the sequence of the amino acids. And then you pick some range of this thing, and that gives you a particular protein. And then this takes me to the last definition of what a gene is. It's a one-dimensional sequence of symbols that codes for a protein. But more than that, because the protein has a unique fold, it codes for a 3-dimensional protein structure. So a gene, in principle, is a 1D sequence of symbols that codes for a 3D uniquely folded protein structure. And then what the ability of biochemists to sequence genes is that we now have large databases that are just filled with these sequences. And these are all of the known genomes that have been sequenced. And now it's so facile and so cheap and quick to do this that we can sequence individual human beings, and we can see the defects in their genetic code, and then we can correlate that with diseases. And so once you have large databases of these things, all kinds of things are possible. So this is actually from New York Times. It's August the 12th, 2022. It says hospital and drug maker move to build vast database of New Yorkers' DNA. And this is, so this is actually, if we can actually build these vast databases, we can use advanced data mining and machine learning techniques to actually come up with therapies and find new drugs and things like that. So the Human Genome Project was enormously successful, and it continues to steamroller on to this day. So something had happened in the mid-2000s, actually around 2010, 2011. President Obama was was the president in the US, and he wanted his own kind of moonshot. And things that were becoming apparent then were some of the problems that we're dealing with were sustainability and also health and so on. And he was somehow persuaded by material science community, I don't know how we did that, but we did, that a challenge for that community is to actually produce, discover new materials and deploy them. And wouldn't it be a great idea if we took the ideas from the Human Genome Project and from genomics and applied it to materials? So this was 2011. This MGI, this Materials Genome Initiative, was proposed, and the goal was to halve the time and halve the cost of finding new materials and deploying them in technologies by using genomic methods, right? And so this is the plot here. Using the conventional approach, this axis is the amount of time, and this is the different phases of the material being developed. And it took this amount of time, and using this fancy genomic methods, we're going to like shrink it down by half like this. And it got everyone excited. So all of these agencies in the US, I mean, I don't think it went outside of the US, but we've got NSF and DOE and the military research agencies and so on, and everyone's very excited about that. So that's Materials Genome, and it's now 11 years after that. So this is a scientific graph, which is a scientific graph which only a politician could love, really. But basically, this is again capturing the same idea that this axis is time, and this axis is number of new materials to market. And using the current approach of materials discovery, which is basically trial and error, and they see it as being linear. It takes a long time and you get a few number of new materials. But if we do this fancy genomics thing and we turn the materials continuum into a loop and we iterate and this and that, and then we'll have like huge numbers of new materials in a very short time. So it's 11 years on, and the question is, has it been a success? And the answer is kind of. Like, if it had really been a success, I think that you would see things like this new therapy or this new battery is a direct result of MGI. Actually, because it's 10 years on, there was an activity to do a new strategic plan, and this was actually a little bit earlier. This was 2019, but this is a paper which is also looking at the success of the MGI. And basically what it shows is, here's one particular example where something rather useful was learned using this circular approach. And here's another example where something was learned. And here's another example where something was learned. So it's kind of a success. I wouldn't say it's a massive success if I was going to measure it. But I think we can forgive it because it came a little bit too early. Back in 2010, there wasn't really powerful machine learning methods that were being widely applied, especially in science. They were just being developed by Google and Amazon and being applied in commerce. But actually, in the next 10 years, we might see some real breakthroughs. And so that's kind of what this new. Oh, this is another example of a kind of success that we have. We do actually have big databases of things. This is the Materials Project database from Berkeley, California. And it has 146,000 materials. Basically, this is completely theoretical database, though. It's all DFT calculated structures and properties. But this strategic plan from this last year, 2021, actually renews this MGI for another 10 years. And they have a new logo now, which still looks circular. And then they call this the friendship bracelet because it kind of looks like a friendship bracelet. But so anyway, it's a new logo, it's a new decade, but now we have machine learning. So the question is, what are we going to do with the MGI? So there's one actually massive problem with the Materials Genome Initiative, and that is that materials don't have a genome. There's no DNA that codes for a material structure. So if we remember what this genome was, it's something to do with DNA, it's something to do with letters. But at the end of the day, it's a 1D sequence of symbols that codes for a 3D structure. And so because I'm a PDF guy, I was always intrigued by the relationship of this to the PDF, because I know Bo loves the 3D PDF, and I do too, actually. But the 1D PDF is I love it even more because it's a sparser representation of the structural information. But yet the question is, can it actually code for a 3D structure even when it's in its one-dimensional form? So this is what I'm going to talk about. So this is the motivation part, and now we'll talk about it. So Bo mentioned PDF. I think many of you know what PDF is, but it's kind of important that you know what it is for this talk. So I'll go through it briefly. And I'm talking about the 1D version. But basically, we have some material. It can be any kind of material. It can be a glass. It can be a crystal. It can be a nanomaterial. It can be a liquid. It can be a solution with something in it. We put it into a capillary. We take it to a synchrotron. You can do this in your lab too, by the way, but we stick it at the end of this pipe, and the X-rays come down this pipe. And then this big thing here is a large area 2D detector, and we simply take the detector and we push it as close as we can to the sample. And that gives us as wide a range of angle on this single detector as we can get. And then all we do is open the shutter and then close the shutter, and that's basically the PDF experiment. So it's very rapid, and it's the X-rays are very penetrating. You can do this in special environments like furnaces and cryostats and so on. And you get circular diffraction patterns on the 2D detector that look like this. And if you have a more crystalline material, these peaks are sharp, they're more like Bragg peaks. And as the nanoparticles get smaller and smaller, these rings get broader and broader, more diffuse, and then when they're really short range ordered or even amorphous, they get to be really broad like this. And what we do is we do an azimuthal integration of the intensity. We do a few other corrections too to get this 1D diffraction pattern, which can be in different forms, I of Q, S of Q, F of Q. And the red line here is actually the bottom one, and the blue line is the top one. And you can see that actually this material, the diffuse scattering is highly reproduced in this material, but what's gone missing are these sharp features that were superimposed on top of the diffuse scattering. In a Bragg experiment, you typically subtract away the diffuse scattering, but this is actually the most favored valuable part of the signal for PDF people. But we do this final step of Fourier transforming this F of Q to get this PDF down here. And the blue one, you see features out to high R. That's because it's a crystalline material, so atomic correlations persist to higher and higher neighbors. And the red one, it's a molecular material, maybe it's cadmium selenide, but it has a correlations out to a certain distance, but then not beyond that because it's a finite nanoparticle system. And then the kind of things we can study you can study anything. You can study, what you pick from under your fingernail if you like. But the things that where the PDF is very powerful is when you have very small clusters. And so some examples that are high impact here are actually these are catalysts that are used in solar fuel production. So sunlight, you want to take sunlight and split water into hydrogen and oxygen. And to do that, you do it in an electrochemical cell, and you have to have catalysts present. And these cobalt oxide nanoparticle catalysts are very good ones. They work quite well, and then we can actually see how large the cluster is, what the internal structure of the cluster is, how well it's stacked, and things like this. This is another attempt by Don Tilley's group at Berkeley to take this cobalt oxide cubane and stabilize it in a reticular network. So this is starting to look a little bit like biology, where you have a complex metal center that's kind of embedded in an organic framework. But this one is actually something that looks a bit like a MOF structure. It's highly porous, but it's basically a molecular ligated cubane. And we can study these even when they are not crystallographically long-range ordered. So these are just two examples of kind of things you can study with a PDF. And you can do time resolved and temperature resolved and in situ and all kinds of things, which I'm not going to talk about today. All right, so to really get to know the PDF, let's look at C60. So basically, this is a diffuse scattering. Through transform it, you get this, and you see a peak at 1.4 angstroms. That's the carbon-carbon nearest neighbor distance. The second neighbor peak is this one on the hexagon. The third neighbor peak is this one diametrically across the hexagon. So that's this peak, and this peak, these first three peaks. And then C60 contains hexagons and pentagons. The pentagon has the near neighbor distance. It has the second neighbor distance, but it doesn't have the third neighbor distance. And so even without modeling, you can gain information. You can see what the bond lengths are, but you can see by looking at the relative intensity of the third peak to the second peak, you can get ring statistics, how many hexagons and pentagons you have relatively. And then if you go, the longest distance you can have here is from this carbon to that one. It's the diameter of the ball, which is 7.1 angstroms. And so the last sharp peak is here at 7 angstroms. And so these sharp peaks down here tell us how the atoms are arranged on the ball. And there's more features out here that are very broad. There's one at 10. And 10 is the ball-ball separation. And then it turns out that actually the balls crystallize in an FCC structure. So the second neighbor peak here will be at root 2 times 10, which is 14. And the third neighbor distance will be root 3 times 10, which is 17. And these are very broad because actually the characteristic function, the shape of the ball, they're basically correlations between carbons on this ball and carbons on the neighboring ball. And the balls are spinning. So data were collected at room temperature. So the correct model for this is a uniform hollow sphere of carbon average carbon density. And it ends up that you get a line shape, which is triangular, which gives us this. But the bottom line is we have these sharp peaks, which are coming from interatomic distances. And so I think many of you know PDF at this point. None of this is a surprise. And when you do a measurement, what you get is a continuous function. It's a continuous function. It's a curve. It wiggles up and down. But my proposition looks a bit like this. If I want the PDF to be a genome for materials, I want a one-dimensional function, but the actual genome is a set of codons. It's a discrete function. It's not continuous. So what we can do is we can actually express this structure here as just the average distance of these atoms, and we'll express it as a number with an arbitrarily high precision. And then the PDF turns into this discrete set of interatomic distances. We call it the distance list. And you can see that there's a multiplicity, like there's actually 90 atoms, 90 interatomic distances with this distance, but there's 60 with this one, and there's 120 with this one, and so on. In principle, if you had a structure that was completely random, so it's a random point cloud, you take Euclidean space, you take a random number generator, and you plonk atoms down inside there, and you don't even have an exclusion zone. You make it so that they can be arbitrarily close. Then the multiplicity is always 1, and you have a large number of interatomic distances. So you can actually express any structure with this distance list. It can have higher symmetry or lower symmetry. And what we have now is we have this discrete 1D function, and it codes for a three-dimensional structure. And so my proposition is, which is a slightly tongue-in-cheek, but my proposition is that the PDF is a possible materials genome. It's a gene for the material. Now, if you take this point of view, like, why would you bother taking this point of view anyway? It's kind of dumb. But if you take this point of view, it gives you a different way to start thinking about the problem. So this is actually something that I did with Phil Duxbury, who is a colleague from Michigan State University. And you can express this thing mathematically as a thing called the unassigned distance geometry problem. So there's a distance geometry problem, which is well known in mathematics. And basically, the distance geometry problem looks a bit like this. You take a graph in the mathematical sense, which is a set of nodes and edges, and if you had a crystal structure, you'd have atoms, and they'd be embedded in Euclidean space. They'd have geometry. They'd have a spatial relationship to each other. But what we do is we collapse it. We flatten it into a graph, and we label each atom. So each atom becomes a node in the graph. And then we make it a complete graph. So we draw an edge between every atom. And then we make it a weighted complete graph where we actually put a value on each edge. And the value that we put on each edge is the interatomic distance. And this can be done in 2 ways. You can have an assigned distance geometry problem where we know the assignment. So we know that this is atom 1, and this is atom 2, and this is atom 3 or atom 4. And we know that this interatomic distance from atom 1 to atom 2 sits here. And so we have the complete graph that's perfectly assigned. We know that it's root 2 between atom 1 and atom 3. Or we can have the unassigned problem where we don't know the assignment. We just have all of the list of interatomic distances that are on the edges, and we have the list of atoms. So we know how many atoms there are, but we don't know how to assign the atoms to the nodes. And that's the unassigned distance geometry problem. And this is what Phil and I worked on. So then the problem comes of how do you solve this unassigned distance geometry problem? So there are 2 steps to the solution. One is to do the assignment, and the other is to do the embedding. And actually, you use the embedding to get the assignment correct, because there's not very many ways that you can assign the atoms to the nodes and it will still embed in Euclidean space. Actually, something that Phil showed was that if you have a random structure that I showed you before, it produces what's called a generic graph. And there's actually a uniqueness theorem that actually the assignment is unique in that case. It's not always unique. But it allows you to come up with kind of algorithms for doing the reconstruction of the structure that look a little bit like this problem. So something we did in 2006 was that we came up with this Liga algorithm where we simply we have a list of distances, and we have, so we can do, for example, the octahedron. So we have a box, we put an atom in the box, we have a distance list, which is 12 times 1 and 3 times square root of 2. These are the interatomic distances in an octahedron. And we simply triangulate new atoms onto existing atoms. If you're in 3D space, in order to place this guy, you need to pick 3 atoms on the cluster and 3 distances from your distance list. And modulo the mirror on this, it will uniquely locate this atom. So then you place it there. And we came up with an algorithm where you had divisions of smaller clusters at the lower level and larger clusters and larger clusters. And you would allow these to compete with each other. And then the high-performing clusters, you'd promote them and you'd add an atom. The poor-performing clusters, you'd demote them and you'd remove an atom. So we had a backtracking method in our algorithm. And it turned out that this Liga algorithm was blazing fast, incredibly efficient. So we could rebuild large clusters just on a laptop computer. We didn't need high-performance computing at all. And the reason was because we were making exceptionally efficient use of the information we had available to us by casting it in this form of this graph theoretic problem. So we can solve a bunch of structures. These were test ones. We took them from crystallographic databases, and yeah, we can solve a bunch of crystal structures. Of course, we didn't do anything there because they'd already been solved by direct methods and by other crystallographic methods, but it just showed that the approach had some value. So since we've solved this nanostructure inverse problem, I should, of course, be very rich and very famous. And unfortunately, I'm neither rich nor famous. So clearly something went wrong. And what went wrong was actually that the Liga would either work or it wouldn't work. And sometimes it worked and sometimes it didn't work. And then it turned out that for a lot of the most interesting problems, it didn't work. So one place where it did work was C60, and this is what the PDF of C60 looks like. And one place where it didn't work, this is a cluster with about the same number of atoms as C60, and it's a very well-ordered cluster, and it's quite rigid. But this is what the PDF of this cluster looked like. And then what you can see clearly right away is that this PDF is much more information rich than this PDF, and it kind of failed on this guy. And so what we discovered is it's basically an information theoretic problem. So to get at this, actually, this work was done by Pavel Juhas, who's my former postdoc and a really smart guy. He's working for Google now. Unfortunately, we couldn't keep him in scientific computing. But he made this plot where he went to a database, a molecular database, actually. So we're taking hundreds and hundreds of clusters from this molecular database. And on this axis, he's plotting the number of atoms on the molecule. And on this one, the number of unique distances on the molecule. So if you have a small number of atoms and if you have a large number of atoms and a small number of unique distances, you have a very high symmetry object. And if you have a small number of atoms and a relatively large number of interatomic distances, you have a low symmetry object. And if you have a completely random object, it follows this n into n minus 1 upon 2 envelope here. And then here the stars are that Liga succeeded, and the crosses are that Liga failed. And what you immediately see is there's a line of death here that if you're above that line, you're lower symmetry and Liga fails, and if you're below that line, you're higher symmetry and Liga passes. And so it's not actually a problem with Liga. Liga is a really good algorithm, regression algorithm. I think it's an information theoretic problem, and that the problem becomes ill-posed. So the way to look at this is actually if you have, this is the information in the PDF data on this side, and this is the degrees of freedom in the model. And if you have a crystal structure, you have a large number of Bragg peaks, and then your model only has to specify a small number of atoms in the unit cell. You tend to be in the well-posed area. C60 was also the case. We had a lot of peaks in the PDF, and then because of the high symmetry, actually you only need something like 3 or 4 parameters to specify the C60. So the degrees of freedom in the model can be low there. But in general, what we're finding is we're in this situation where the information in the PDF, as the symmetry goes down, the information in the PDF goes down, and the degrees of freedom in the model tends to go up because the symmetry has gone down. So when these two cross, we go from a well-posed problem to an ill-posed problem. And then we need to find very creative ways of solving ill-posed problems. We have to add information on the input side, or we have to remove degrees of freedom on the output side. So what's this? This is in the, okay, yeah, I remember what I was going to do here. So this is an example here. I have a very strong collaboration with Kristin Jensen, who's in University of Copenhagen. She's a former postdoc. She's doing wonderful work in Denmark right now. And she formerly worked with Bo, actually, so there's a connection. But she is actually, this is one kind of example of something that might trip us up. So the data here is that we've got a solution of ammonium metatungstate hydrate, which goodness knows what that is. I'm not a chemist, I have no idea. But basically what she tells me is it's a bunch of inorganic tungsten oxide clusters that are floating around in solution. And when you measure it, the PDF looks like this top blue line here. And you can see this top blue line looks a bit like that cadmium selenide cluster line. There's some broad peaks, there's a few of them. And in this case, we know the right answer because Kristin's students had actually solved it previously. And it's a alpha Kegin cluster, which is W12040. But the key thing is that the PDF looks like something that LIGA would fail on. And so one thing that you can do is you can take degrees of freedom out from the model, and this is the approach that we've been doing with Kristin. And so LIGA is a cluster constructor algorithm where we build up clusters just using the information we have in the PDF. And then the insight that Kristin had was that what we could do is actually maybe have a cluster deconstructor algorithm. So we can give it a plausible structure that we could pull from a crystallographic structural database. That's plausible because it actually formed in nature. And then what we can do is we can come up with an algorithm that removes atoms from that cluster in such a way to get a good agreement with the data. And so the reason she solved that Kegin problem was that she had used her chemical intuition and then actually used that approach in a brute force way. But it turns out that when the clusters get large, this becomes very inefficient. There's a combinatorial explosion and you can't really do it. So working also with Raghav, who's a computer scientist and a machine learning guy in Denmark, and this is Andy Anker and Emil Kjær, who are students. We came up with a motif extractor, which is this cluster deconstructor. So the algorithm goes a bit like this. You start with a large set of known structure fragments, which you get from crystallographic databases, and you use a cookie cutter to like stamp out a finite size cluster from the crystal. You can make that as big or as small as you want, or whatever shape you want. But then what you do is you systematically prune or remove atoms in such a way as to improve the agreement with the measured PDF. And you iterate over the set to find a global solution, or in principle, you can get a set of reasonable solutions. And so this was done, and it worked. It's brilliant. So these are different kind of superclusters, including blue and yellow atoms. And then the yellow atoms are the ones that the algorithm retained, and the blue atoms are the one that it threw away. There's something gone a bit wrong here, but this one's right, this one's right, this one's right. And so basically it's giving basically the right answer in an automated way. And I think this is going to be a very powerful approach moving forward because we have crystallographic databases, and so we can prune them. And so even when we have kind of low information content PDF data, we have a way forward to do it. This actually used a machine learning model. It was a gradient boost decision tree. And one of the nice things about this was that it speeded up the modeling. So the way we would do the modeling normally is we would compute the PDF for each different candidate structure and then convolute it to get the thermal factors, and then extract the RW, which is the weighted agreement factor. And what the machine learning model was able to do is that it was able to learn or predict what RW it would get for a given structure. And then it was able to extend so that if you gave it a structure it hadn't seen before, it could predict the RW without actually doing the, without actually computing the PDF. It actually worked quite well. It gets things wrong, of course, but it's actually kind of helpful. Okay. So the mention of machine learning is apropos because actually something that machine learning is very good at in general is actually solving ill-posed inverse problems. And the way you do that is you give it a huge amount of data and huge history of data to learn from of plausible solutions. So machine learning is inherently looking a little bit like that motif extractor, cluster deconstructor. You let it learn on large numbers of plausible structures, and then you let it kind of filter through them to get an answer. So we tried using very standard conventional convolutional neural net to actually solve the problem of, if I have a PDF, can I tell the space group of the material it came from? So the input is just the PDF. So if you ask a human being to look at a PDF and ask them to say which space group was the material in that gave that PDF, they'll have a very hard time. They might recognize it if it's FCC, because we've all looked at FCC PDFs a lot, but in general, it can't be done. But in practice, you know that the information is in the PDF because the information is in the diffraction pattern, and the PDF is the Fourier transform in the diffraction pattern. And so the question was, can we train a machine learning model to do that? And then the answer was yes, it worked actually quite well. It's a fairly conventional convolutional neural net. And we had 100,000 structures in the inorganic crystal database that were in 45 most common space groups. We didn't try and classify into 230 space groups because many of them are very sparsely populated. So we split it into an 80-20 training, validation, testing set, and we trained it. And long story short, we get, if our success definition is that the right space group is in the top 6, we get it with 90% precision. It works slightly better for the higher space group numbers. These are the high symmetry, more like cubic ones up here. But for the highly populated, low symmetry ones, it does quite well, and then it gets it wrong for some down here. It's really also interesting about how it gets it wrong. So this is the matrix of confusion. The matrix of confusion for the rows, you say which space group did the classifier classify into, the predicted space group, and on the across the top here you have the actual space group. And if the classifier was working perfectly, this would be a diagonal matrix with ones along the diagonal. And what you can see is, largely speaking, it actually works really well. But interestingly, there are these teardrops here where certain actual space groups get misclassified into a bunch of other space groups down in this vertical column here. And actually, it turns out that there's physics behind that. It's actually related to the group subgroup relationship of these space groups. And this is actually something that was known before the machine learning was done. It was actually known from conventional indexing that the indexing programs get confused in this way. And so actually the machine learning was teaching us physics by getting things wrong, which was kind of interesting. What we would love to do too is to use machine learning to actually do structure solution. And this is a bit more difficult. And again, with the same team, we've been trying to do structure solution, in this case using a convolutional variable autoencoder. And so with this one, you have a high dimensional input space, which is the actual 3D structure, and you encode it into a low dimensional latent space. And then you run it through the decoder and it puts it back into the higher dimensional space again. So you start with a structure, you encode it into a low D space, and then you decode it into a high D space. And this is not dissimilar from doing principal component analysis or something like that. where you have a large set of inputs and then you find the principal components and then you reconstruct the outputs as linear combinations of the principal components. So it's kind of very similar to that. But what we do is we also give it as an input the PDF. And so each of these points in this latent space is a structure, but it also has with it, it's labeled with a conditioning PDF. And what that means is that we can run the autoencoder in a different way, where we give it, later on, we give it as an input, the PDF, and it can find the point in latent space where that PDF came from. And then it can decode that and give you back a structure. And if the PDF is one it's seen before, it will give you back the right structure. But if it's one it hasn't seen before, it will land in this latent space in a gap. But it can do interpolation somehow, and it can then give you back what it thinks is the right structure. So it's a generative model. It can produce outputs that it's never seen before. So anyway, let's see how it does. Well, because it's basically doing interpolation, what we want to do is we want to run it on something where we can get the right structures by interpolating. And the close packed metals were the right ones to do that with. Because actually you can interpolate between FCC and HCP with stacking faults, for example, and you can interpolate into decahedral structures which have also stacking faults or twin boundaries in, and so on and so forth. And long story short is it actually worked really well. And so it kind of works, although it's very limited in scope right now because it's just looking at close packed metals. So I'm basically out of time. I just quickly want to give some vision for the future that in principle, if we can capture the databases that we have right now are mostly theoretical in materials. And the few experimental ones are things like crystal structure databases. And we're doing a ton of stuff with the crystal structure databases. But what if people were doing experiments and they were capturing it all into a database? We could do much more. And we could come up with modeling procedures. This is something that Soham, my previous student, did, where instead of actually picking a single structural model and fitting it to the data, you can pick, you can trawl through large databases of structural models and do model selection and compare them to the data, and you can separate. These were, again, the FCC clusters, and we found that the decahedral model was working much better for these palladium nanoparticles than the conventional modeling. And if you have databases of crystal structures, which we do, we can simply upload a PDF and it can tell you what the structure was from the database directly. So these are the kind of things we can do if we could just capture experimental data. And so that's a big effort in my group right now, and I'm trying to persuade the community to move in that direction, to share their data because there are sociological issues that we can discuss. But this is pdfitc.org, which is a website where you can upload a PDF and get a structure back. You can upload a PDF and get a space group back. It also has some unsupervised machine learning methods like non-negative matrix factorization, where you can upload a set of data and it will give you back the principal components, basically. And so this, I think, is the future. And then even more the future, this is a project of Martin Carlson, who's another Danish visitor we had in the group, and Barak Oza, who's a group member. And actually, we want to go in the direction of a machine-readable scientific literature. So right now, the scientific literature is written by humans for humans to read. And what I'd like it to be is firstly written by humans for humans and machines to read. And secondly, written by machines for humans and machines to read. So then it makes my life much easier. But this is the initial steps of this is actually in collaboration with the IUCR. And here's a paper in Actor A which describes it. But basically what we do here is you can upload a powder pattern and it will give you a rank ordered list of papers from the IUCR database that might be relevant for to your pattern. So you can start actually doing literature search very early in your study. So just to summarize then, PDF is a list of interatomic distances in a three-dimensional atomic structure. The PDF is therefore a 1D discontinuous function that codes for the 3D atomic structure through the UDGP, the unassigned distance geometry problem. And it therefore resembles a gene or a genome, and it's a material genome. It's not like a gene in the sense it doesn't drive the structure. but it is like a genome in other respects, and it has an advantage as a representation for structures, that it's universal. It doesn't require the structure to be crystalline. And so in this talk, I've explored some things where this could take us in principle. So thank you for listening. I'd like to say thanks very much to the students and postdocs and visitors who do all this wonderful work, and the facilities where we do the work, and the funding, of course, and wonderful collaborators. And yeah, thanks very much, and I'm happy to take questions.
Thank you. Krzysztof, you are unmuted.
Thank you very much, Simon, for this brilliant lecture. I can envisage situation when machines start competing for grants with us. So we are on the way.
We need a machine that gives us grants. Yeah.
Anyway, the next now for comments and questions. Before they appear, could you comment on this kind of idea? I mean, do you think that this approach could be extended to other fields? For example, imagine that we analyze universe with PDF, distribution of stars or galaxies, looking for some hidden order behind this. What's your opinion about this kind?
Yeah, it's really funny. It's really funny that you mentioned that, because that's actually already being done. So there's actually work in cosmology of taking star maps and then extracting different invariants, including exactly the PDF of the stars. Yeah. So absolutely, yeah. I mean, so it's you know, we've been for 100 years or 100 and however many years, 110 years studying structure, and we've always had a very kind of crystallographic point of view, and you know, space groups and symmetry was like a really enabling technology that came along, but I think it got us a bit stuck, because now we're kind of stuck in kind of symmetry space group crystallography land, but actually a lot of the richness is then a little bit lost. So if we had different representations for structures, and this is one of them, I think it could open up different ways of thinking and ways of, you know, different ways of being creative. Yeah.
And also transformation. I mean, we mostly stick to only a few transformations, Fourier transform, etc. Whereas the number of mathematical possibilities is really infinite almost. So maybe
that's true, but the Fourier transform in this case is particularly helpful because it does map the diffraction space into the crystallographic real space. That was shown by Zerniger and friends, right? That the autocorrelation of the density is the Fourier transform, the diffraction data. So we can explore different transforms, but we'll be transforming to things that are not well defined, not quite so well defined. So I'm not sure, but you know, this I think it's an interesting idea. It's something that should be thought about for sure.
Yeah.
But if you think about the PDF as a genome, you can also think about incorporating defects in materials. And then one of the most exciting things that happened in genomics was when it became sufficiently quick and inexpensive to sequence genes that it wasn't a 15-year project to do one human genome, that you could do an individual person's genome in, you know, in 15 minutes. And now we have 23andMe, and we have, you know, all of these things of sequencing individuals. And what you're then looking at is you're looking at the defects in the genome,
yeah.
and you're mapping that onto the individual. And of course, we can do that in materials because it's actually the defects in materials that make them interesting. So in principle, this way of thinking could help us to map, you know, we could have databases of defects and things like that.
Paulina, there is a question from Hans Beth.
Yes, he's asking, are there uniqueness problems using your approaches from 1D PDF to 3D structures? Remember this problem in interpreting Patterson functions?
Yeah, so that's a great question. Thanks, Hans-Piet. So we think about this quite a lot, and there's two answers to this question. I want to try and separate these two answers because people muddle them up. One of them is if you have your PDF with infinite precision, is there a unique 3D reconstruction or not? So what we know is that if you have a random structure, there is a unique reconstruction. But if you have internal symmetries, there may be a unique reconstruction, but there may not be. We know of examples where there aren't. So it turns out that if you take a hexagon, there are actually 3 different 3D structures that give the same PDF, or there's 2 3D structures that give the same PDF of the 2D hexagon. So there are 3 arrangements of points in space that will give you the same PDF in that case. So that's absolutely not unique. But what we found is that actually uniqueness is not our biggest challenge here. That Richard Welby, for example, worked really hard to find structures, two structures that had the same pair correlations but were different structures. But he had to work really, really hard. So these are a kind of interesting edge cases, but they don't concern me. But then the question is, well, in that case, why aren't I rich and famous, right? And the reason I'm not rich and famous is because when we measure the PDFs, we measure them with finite resolution. So we don't get that discrete set of distances. What we get is the continuous function that goes up and down. And then what happens is you have huge information loss. If you measure a random structure with finite resolution, you just get a horizontal line. And so the problem is information loss in the measurement, much more than any other aspect. But it's really that information loss that is really kind of hurting us in terms of this uniqueness of the solutions. So that would be what I'd say to Hans-Piet.
Vitaly Kurlin is asking, is it true that PDF cannot distinguish homometric crystals known due to, yeah. And there is a citation given.
Yeah, this is the thing this is what I just addressed.
Exactly.
Yeah. So this is absolutely true and absolutely, in my view, kind of irrelevant.
From Bo Hao Chen. Hi, I'm Bo Hao from Taiwan. I'm curious about the distance resolution of PDF data. How detailed the data could PDF investigate when we try to reconstruct the structure or model?
Yeah, so I addressed this resolution issue that if we're measuring the distance list, but we're measuring it with finite resolution. So the question is, how can we do better? Like, how can we do a higher resolution? So what I'm talking about with resolution, I'm talking about real space resolution. And the real space resolution is given by the Q range, the range of reciprocal space that you measure over. So we can measure to higher Q. But this runs out at some point because if we go to sufficiently high Q, you get all of the information that's available to you. And if you go to even higher Q, you're just measuring noise. So very often we're actually going to sufficiently high Q. And then the resolution is basically determined by the thermal motion. And so then you can go to low temperature to reduce the thermal motion. And if you go to 0 Kelvin, you still have quantum 0-point motion. And that's the kind of fundamental universal limit on the resolution that we can get in the PDF of a real material. And unfortunately, it's quite, even that case, it's not that great. It's going to make it hard for a lot of materials.
Hans-Bert is asking if you add the temperature-dependent data. To what extent?
Yeah, this is a great point.
Yeah.
So when I was talking about the ill-posedness of the problem, there's two ways to tackle it. You remove degrees of freedom from the model, or you add information on the input side. And this is one example where you can add information on the input side. And it works quite well. So that's a great point from Hans-Bert. We actually have done a little bit of work recently. We've been working with this unsupervised learning, machine learning method, non-negative matrix factorization for extracting. If you have an in situ reaction happening, it will show you the components as a function of time during a reaction in a very beautiful way, actually. But we've played around with actually looking at temperature-dependent data of a sample which had two phases in it. And we can actually extract the phases using NMF on the temperature series by developing a stretched NMF algorithm that removes the thermal expansion part. So that's a particular example of the general point that Hans-Bert is making.
Vitaly Kulin is mentioning that the non-uniqueness problem has been resolved by point-wise distance distribution, which is stronger than PDF.
Yeah, this is true. So I talked to Vitaly at ECM, and his work is very exciting. It's definitely worth a look, and we're certainly hoping to work with Vitaly to take that forward. So my way of thinking that I presented in this talk is actually Vitaly's been thinking along these lines also for the last 2 years. I was not aware of it, or last few years, I was not aware of it, and then we met at ECM. So this is very exciting. I have some friend out there who's also not a space group person, and I hope we'll be able to work together. But yeah, he's got a different metric, which is an invariant, which is stronger than the PDF.
Actually, the same problem is discussed in protein world and macromolecules, right? How to represent, and distance kind of information is being replaced by some different, more sophisticated representation, right?
Yeah, you can start putting in kind of higher order modes. So they've been thinking about it also for a while. But we can take it the other way too. We can take what we're doing and we can apply it to proteins as well, because proteins are just materials, it turns out. In fact, you are just a material, Paulina. I hate to tell you.
Yes.
But you're a very interesting material. You're not a boring one like diamond.
Regarding the space group, could it be like your approach discovered that some materials have been assigned the wrong space group?
Yeah, I think actually Vitaly has actually examples of that. He might be able to tell us. Yeah, absolutely.
Imagine that we could identify in which space group do we live in this universe.
Yeah. Well, I think it's mostly P1, isn't it?
Is it? We do not know this. And how about parallel universes? Anyway, do we have some more questions or comments? My impression is that Bo has a question, but no. Okay. Anyway, thank you very much for those excellent, really wonderful lectures.